Hello and welcome to everyone joining us for today's Drug Target Review webinar titled Reprogramming Autoimmunity, Designing Smarter Checkpoint Receptor Agonists. This webinar is brought to you in association with Eurofins DiscoverX. My name is Dr. Carrie Haslam, Editor at Drug Target Review, and I'll be moderating today's session. I'm delighted to be joined by Dr. Daniela Cipolletta, Senior Director of Immunology at Seismic Therapeutic, to explore how immune checkpoint dysfunction drives autoimmune disease. She'll explain why PD-1 agonism offers a promising route to controlling T-cell activation when applied correctly, and how S4321's unique dual cell design delivers sustained immune regulation without depleting protective T-cells. Daniela will also unpack the unexpected role of low affinity binding in maintaining immune balance. Following the presentation, we'll move on to our question and answer session. So please feel free to submit your questions at any time during the webinar via the questions panel on the right-hand menu. And with that, I'll hand over to Daniela to begin the webinar. So over to you, Daniela.
Hello, everyone, and thanks to the Eurofins team for the introduction. Thanks also for the opportunity of sharing how at Seismic we have designed smarter checkpoints receptor agonists to reprogram autoimmunity. And before going deep into the data, I would like to first address what at Seismic we consider as a smarter approach. For instance, biologic targeting multiple pathways and/or multiple types simultaneously have the potential of restoring immune homeostasis. And this is simply because pathogenesis of many autoimmune diseases are driven by cell-cell interaction. And one of the interaction that is best characterized is the one between a T cells and an antigen-presenting cells or a B cells. Therefore, at Seismic, we have generated dual cell bidirectional agonist antibody targeting simultaneously multiple inhibitory receptor. And this is really for the treatment of dysregulated cell-mediated immunity. The reason why we have focused on inhibitory receptors is because inhibitory receptor deficiency has been demonstrated to be associated with autoimmunity, not only in mice, but also in humans. And a few examples are provided in these slides. For instance, PD-1 deficiency in mice leads to an accelerated onset and an increased incidence in type 1 diabetes, as you can see here on the top. And CTLA-4 deficiency in mice leads to a massive lymphoproliferative disorder. Lymphoproliferative disorders is also what is observed in clinical cases of human CTLA-4 haploinsufficiency. As you can see here from those CT scan images, there is lymphocytic infiltrates that have been observed in non-lymphoid organs. This is together with CD4 lymphopenia and a decrease in circulating B cells and an overall increase in PD-1 expression on CD3 that are T cells. Clinical cases of PD-1 deficiency are similarly characterized by type 1 diabetes, hypothyroidism, and juvenile arthritis. And as you can see here from this image, there has been observed that the serum from those patients are autoreactive towards antigens that are associated with thyroiditis, with diabetes, and pneumonitis. And it is possible to target checkpoints receptor in autoimmunity because both activating and inhibitory receptors are highly expressed. And while it's very easy to explain why an activating receptor is overexpressed during a condition, an inflammatory condition, it is less logic to explain the or the overexpression of an inhibitory receptor. What is thought, in fact, is that inhibitory receptors are highly expressed in autoimmunity, likely as a natural mechanism to try to restore normal immune homeostasis. What is illustrated here in this figure is the expression and distribution of several checkpoints receptor in a case of lupus nephritis, where you can see that checkpoints receptor are really enriched, especially in the T cell compartment. Therefore, they can be targeted. But the role of inhibitory checkpoints receptor in autoimmunity and in maintaining, in general, immune homeostasis, I think is really well exemplified by the new occurrence of immune-related adverse events, which occur in cancer patients when they are treated with checkpoints antagonists. Illustrated here in these figures are acute and chronic immune-related adverse events across a different range of organ systems. This really underscores the role of inhibitory checkpoints in maintaining normal immune homeostasis, and I think it's worth it to note that it has been observed an increase in frequency and the earlier onsets of immune-related adverse events when IO therapies are combined. This really suggests that inhibitory pathways are actually playing a non-redundant role. So therefore, the therapeutic hypothesis we follow at Seismic is this one. If loss of blocking of inhibitory receptor results in autoimmunity or inflammation, agonism of these pathways might restore normal immune homeostasis. And we decide to target PD-1 as an inhibitory receptor. And why? Because PD-1 agonism, we knew at that time when we started, we knew that it can be accomplished by the use of antibodies. We also knew from the work of our peers that strong agonism requires superclustering. And one way to achieve receptor superclustering is through the use of antibody capable of anchoring to Fc gamma receptors. Fc gamma receptors are expressed by antigen-presenting cells, as illustrated here. And what that means is that no or weak PD-1 agonism is observed when a bivalent antibody binds PD-1 and simply cross-link PD-1 without inducing superclustering. And high-order superclustering of inhibitory receptor by antibody is instead really what is necessary to deliver a strong agonism of PD-1. Unfortunately, it's not very easy to decide and to generate antibody that targets selectively Fc gamma receptors. And the reason, I think, is well illustrated by this slide. The Fc gamma receptor family, like checkpoints receptor, also come in different flavor. There are activating and there are inhibitory receptor. In reality, I should say, there is only one inhibitory receptor, which is Fc gamma receptor 2b, while all the other Fc gamma receptors are activating. And the challenge in targeting members of the family is that there is high degree of sequence identity across them. For instance, between Fc gamma receptor 2a, which is activating, and Fc gamma receptor 2b, which is inhibitory, there is about 95% of sequence identity. And if we choose an IgG1 wild type, for example, as a backbone of a therapeutic antibody, for example, an agonist antibody, we know that we will not discriminate between binding to the activating or binding to the inhibitory receptor. And just to give you an idea, a binding to an activating receptor can trigger pro-inflammatory cytokine, while the binding to an inhibitory receptor instead limit pro-inflammatory cytokine production. Now, when we start this program, we knew that all the first generation of PD-1 agonist antibody were in an IgG1 wild type backbone. So we saw this as an opportunity to generate a second generation or novel and improved therapeutic antibodies by targeting selectively Fc gamma receptor 2b. So we are aiming to restore immune homeostasis by agonizing multiple inhibitory pathways with one antibody that we call dual cell bidirectional antibody. The reason why we call dual cell bidirectional is because two cells are important for this antibody to work. This antibody will bind with variable region PD-1 on autoreactive cells, but the agonism of PD-1 is supported only when the same antibody binds selectively with Fc, the inhibitory Fc gamma 2b receptor. By being selective for Fc gamma receptor 2b, we will avoid binding and activating Fc gamma receptor 2a, and therefore leads to the production of pro-inflammatory cytokine. By being selective for Fc gamma receptor 2b, we also will avoid activating Fc gamma receptor 3 or CD16, which are the main receptor that trigger ADCC, antibody-dependent cellular cytotoxicity. So the generation of antibodies that target selectively Fc gamma receptor 2b is known to be very challenging. And I think the degree of the challenge is well illustrated by this overlaid structure of representing the similarities between Fc gamma receptor 2a and 2b in magenta, while in orange you can appreciate how few are those differences between 2A and 2B that can be used to generate a selective binder for Fc gamma receptor 2B. We were able to solve the puzzle by utilizing our IMPACT platform. Our IMPACT platform is an integration of different capabilities. For instance, an integration of machine learning with structural biology, protein engineering, and finally, translational immunology. Thanks to our machine learning capabilities, we were able to generate several Fc gamma receptor 2b selective Fc that were then pressure tested in a screening funnel, as you can see in this slide. And the first things we evaluated on our Fc moieties is their affinities for the inhibitory receptor, Fc gamma receptor 2b, and activating receptor, Fc gamma receptor 2a. And as you can see from this graph, we really identify Fc moieties with a varieties of affinity to both Fc gamma receptor 2a and Fc gamma receptor 2b. But we really did not rely only on affinities measurements in order to really assess and pressure test selectivity of our molecule. We, in fact, tested all our moieties in a counter screen assay, which is a cytokine release assay. In this assay, we utilize primary human dendritic cells that are derived from monocytes. Those cells will express both Fc gamma receptor 2a and Fc gamma receptor 2b. Those cells are activated and cultured in plates that are coated with the antibody that we want to test. Now, what is the expectation is that when we coat on the plate a human IgG1 wild type, we will observe an induction of TNF-alpha, as you can see here from the first bar. And also there is the expectation that if we instead coat on the plate a Fc gamma receptor 2b selective Fc, this induction is instead abrogated. And all here in light gray, you will see the behavior of seismic novel Fc gamma receptor 2b mutants. Some definitely better than others. So we are really pressure test selectivity thanks to these primary assays. And now it was really important for us to verify one more thing before selecting our Fc gamma receptor 2b engager. The last thing that we want to check is the capability of those Fc to support PD-1 agonism. And this is where Eurofins really came of a huge help for our program. In fact, to assess how seismic Fc gamma receptor 2B selective Fc design supported PD-1 agonism, we adopted and tweaked Eurofins DiscoverX PD-1 antagonist assay. Before going to the details of how the assay works, I think it's important to mention what is really the expected biology that we were trying to replicate thanks to the use of an in vitro assay. We wanted to observe and measure inhibition of T-cell activation. And that inhibition is really the result of an interaction between an antigen-presenting cells and a T-cells. It really occurs at the synapse between those two cells, as illustrated in this slide. So antigen-presenting cells express the ligand of PD-1, which is called PD-L1, while the T cells carry the receptor for PD-L1, which is PD-1. What happens is that upon PD-L1 binding to PD-1, phosphorylation of tyrosine residues within PD-1 cytoplasmic region occur. And this event is necessary for the recruitment and activation of SHP2. Recruited SHP2 mediates dephosphorylation of proteins that are associated with the T-cell receptor, such as ZAP70. But also, SHP2 inhibit co-stimulatory molecules that are very important during the street signaling transduction, such as CD28. So these events attenuate TCR signaling strengths and ultimately translate also in a decrease in cytokine secretion, such as IL-2. Now, the Eurofins DiscoverX assay, PD-1 antagonist assay, really mimic this biology by utilizing a U2OS cell line which overexpress PD-L1 or PD-L2. And this cell line is co-cultured with a Jurkat PD-1 line, which is engineered to measure the recruitment of SHP2 to PD-1 through the generation of chemiluminescence signal. This signal results from a complementation of two beta-galactosidase enzyme fragments, one that is fused to the PD-1 and the other that is fused to SHP2. Therefore, when SHP2 is recruited to PD-1, the enzyme becomes active. With the presence of a substrate will produce light, and this is used as a proxy of PD-1 agonism. Now, in the Eurofins assay, the use of an antagonist for PD-1 that interfere with the interaction between PD-L1 and PD-1 will block the generation of light or luminescence. And this is what is called PD-1 antagonist assay. Now, we tweaked this assay in order to measure PD-1 agonism. How? Instead of utilizing a U2OS cell line that express the ligand PD-L1, we're utilizing what we call an Fc gamma receptor 2B anchoring cell line. So this cell line express Fc gamma receptor 2B, and the expectation is that our antibody will bind Fc gamma receptor 2B and therefore promote PD-1 agonism exactly like the natural ligand PD-L1. And we were very pleased with the results obtained by utilizing the Eurofins DiscoverX PD-1 and SHP2 reporter line, because it allows us to develop not only a high throughput in vitro system, but especially a robust screening assay with a wide dynamic range to measure PD-1 agonism. So again, what we have done is to co-culture a Fc gamma receptor to be expressed in line with a T-cell reporter line, the Jurkat PD-1 SHP2. And we utilize to pressure test this system prototypes antibodies. Those are antibodies that are agonists for PD-1 and carry either an Fc that is selective to Fc gamma receptor 2B or an Fc that binds, that is an IgG1 wild type. We also have mirroring control, which are antibodies that do not bind PD-1 but bind RSV and should not be active in the system. These antibodies are used to understand the background level in our assay. As you can see from this graph, we really noticed that the first things that we learned was that anchoring our antibody to Fc gamma receptor 2b was providing a stronger PD-1 agonism compared to the antibody that are in an IgG1 wild type. So this is the difference between the teal curve, that is a 2b specific antibody, versus the gray curve, which is an IgG1 wild type. But what was very interesting is that if you zoom in into this, into the gray curve, you can still appreciate that there is agonism. It's just not as strong as the one provided by Fc gamma receptor 2b anchoring antibody, but you can still measure, which is an important point when you are developing such a sensitive assay. And to ensure that our assay was not too far from physiological conditions, we have then substitute the Fc gamma expressing line with human PBMCs. And we were really pleased to see that, again, Fc gamma receptor 2b selective PD-1 agonist antibody anchored to primary cells elicited a superior PD-1 agonism in our T-cell reporter line. And we were able, therefore, to confirm the data that we generated by utilizing the cell line. And it was really only at this point that we felt comfortable to screen our machine learning derived FC moieties. And we were observing that Seismic FC design, the one that lacked pro-inflammatory cytokine production, were still able to maintain and support PD-1 agonism. As you can see here, we identify FC that support agonism very similarly to an IgG1 wild type, but also FCs that actually enhance PD-1 agonism compared to an IgG1 wild type, or that are a little weaker compared to an IgG1 wild type. So definitely a variety of behavior here to choose from. So until now, I really explained how we identified our Fc gamma receptor 2b engager and how important was machine learning to design those molecules. But we were able to identify our PD-1 agonist by utilizing a different approach, which is a conventional antibody screen. So from our antibody, classical antibody campaign, we identified several PD-1 agonist antibodies that were not antagonists. We test both antagonism and agonism thanks to the Eurofins cell lines. And in the antagonist assay, we utilized U2OS cells expressing PD-L1 in co-culture with the Jurkat PD-1 SHP2 reporter line. As you can see here, there was really no change of SHP2 reporter activity in presence of our Seismic PD-1 antibodies. And this is really a striking difference from the behavior of an antagonist antibody represented here with the black curve that decrease SHP2 reporter activity in this assay. All our PD-1, Seismic PD-1 antibodies were instead agonists and in fact increase SHP2 reporter activities to different degrees. That means that we were able to identify a range of functional agonistic activity. To prove that PD-1 agonism was truly supported by the binding to Fc gamma receptor 2b, we knock out Fc gamma receptor 2b from our anchoring cell line. And when the Fc gamma receptor 2b knockout cell line was then co-cultured with Jurkat PD-1 SHP2 reporter line, we did not observe, as you can see here, any agonism. We then concluded that our panel of dual cell bidirectional PD-1 agonist antibodies were not antagonists and relied on anchoring to Fc gamma receptor 2b to agonize PD-1. We discovered a novel PD-1 agonist and Fc gamma receptor 2b dual cell bidirectional antibody by integrating machine learning approaches with conventional antibody screen. And what was really instrumental for the selection of our development candidates, which we call S4321, was the variety of affinities, activity, and epitope of the moieties that we generated. And I will tell you more about how important was especially the affinity into the selection of S4321 as our development candidate. The PD-1 agonist component of our development candidates, again, that we call S4321, is in fact low affinity and as an activity for PD-1 that is similar to the activity of the natural ligand PDL1. As you can see here in the PD-1 agonistic reported assay, the orange coded molecule S4321 is more similar to PDL1 FC compared to other molecules that appear in this graph. One is a benchmark in gray, peresolimab, and the other is the high affinity version of S4321, which is a molecule that differs from S4321 only for one amino acid. The mutation compared to high affinity and a stronger activity in this reporter assay. It is very important to note that all our selected agonists were not antagonists in our assay. And you can see here there is really no change in the SHP2 recruitment activity compared to a true antagonist, which is nivolumab. So why we selected a low affinity agonist with similar activity to the natural ligand PDL1? And the reason is really illustrated in this slide. It was really one of our first observation in what we observed through the use of whole blood culture and time course experiment is that S4321 low affinity variable region does not alter PD-1 expression on T cells. And this is in contrast to high affinity antibody, such as here the blue label or the gray labels that cause loss of PD-1. And in order to verify what was the functional or the biological consequence of losing PD-1, we repeated the same assay by adding an antigenic stimuli, which is represented by SAB. And what we observed, again, is that S4321 low affinity variable domain still preserved, also in this context, the expression of PD-1 on the surface of T cells in contrast to the high affinity antibody, but especially the S4321 maintained the control of ALK2 production by activated cells. And this is in contrast to nivolumab, for example, which is expected, but also the high affinity antibodies, such as our own, the blue molecule, the high affinity version of S4321 and peresolimab. And in order to verify that this was really a universal applicable concept, we took all the benchmarks that were available to us that are characterized by high affinity. And again, we verified that low affinity antibody binding correlates with maintenance of PD-1 expression, as you can see here. And the maintenance of PD-1 expression correlates with ALK2 production. So loss of PD-1 is now correlated with induction and production of ALK2 from high affinity antibodies. The observed results from our in vitro culture were recapitulated by utilizing in vivo animal models. Here, we are measuring PD-1 expression levels on blood-derived T cells, and upon administration of 2 test articles in a dose response in the humanized PD-1 knock-in mouse model. One test article is the high affinity PD-1 agonist antibody peresolimab, here illustrated in gray. The other test article is S4321 and illustrated in orange. As you can see, peresolimab induces a very rapid loss of PD-1 expression on T cells already at day 1, while PD-1 levels are maintained in S4321 treated animals. And it is worth to note that PD-1 levels returns with peresolimab starting from day 7 because the exposure is lost. And a recent academic publication supports seismic hypothesis that high affinity antibody binding drive loss of cell surface PD-1. The publication shows that bivalent anti-PD-1 antibody binding on CD8 T cells cause PD-1 endocytosis and therefore loss of a cell surface PD-1. This is basically the PD-1 internalization enhance T cell effector function and should be considered as an additional mechanism of action of PD-1 antagonist antibodies. These results are consistent with our own observation that high affinity bivalent, now PD-1 agonist molecules also result in an apparent loss of Blake. And this loss of Blake leads to the exhibition of a behavior that is similar to the one associated with an antagonist antibody. So what I've shown so far is that S4321 differentiated from other approaches by agonizing PD-1 with a low affinity variable domain. But our molecule is also differentiating because simultaneously agonize Fc gamma receptor 2b. As you can see from the panel here on the left, S4321 agonize Fc gamma receptor 2b in a SHIP1 recruitment assay. In this assay, only S4321 and not the other benchmark on the bottom were capable of inducing recruitment of SHIP1 to Fc gamma receptor 2b, which is used as a proxy of Fc gamma receptor 2b agonism. Furthermore, by binding selectively to Fc gamma receptor 2b, S4321 does not elicit the production of pro-inflammatory cytokines such as TNF-alpha from antigen-presenting cells. And this is unlike an IgG1 wild type, here in gray, that induces the production of TNF-alpha because it binds to activating receptors. And last but not least, our Fc does not activate Fc gamma receptor 3 and therefore does not mediate ADCC and depletion of T cells which are PD-1 positive, or in particular Tregs that are PD-1 positive. And here on the right, we show that S4321, which is in orange, does not deplete PD-1 positive Tregs, unlike the first generation of PD-1 agonist antibody, peresolimab and rosnilimab, which do deplete T regulatory cells. Not only we don't deplete Tregs, but S4321 promotes the induction of Tregs in vitro when we culture naive CD4s in presence of TGF-beta and IL-2. And we believe that S4321 recapitulated the reported observation made with the natural ligand of PD-1, which is PD-L1. And in fact, it has been demonstrated that PD-L1 promotes the differentiation of iTreg not only in vitro, but also in vivo. It has also been demonstrated that PD-L1 sustains FOXP3 expression and promotes a stronger Treg suppressive function. And we were very excited when we saw that S4321 exhibits efficacy across a number of parameters in an acute model of graft versus host disease. In this model, we noticed that S4321 treatment reduced splenomegaly. It also reduced the expansion of donor cells, as you can see here. But what really made us super excited about this data is the observation that S4321 was efficacious at reducing pro-inflammatory mediators, not only those ones that are produced by T cells, such as interferon gamma or IL-2, but also those produced by myeloid cells, and such as IL-10 or MDCs. And why this was really important for us is because those changes observed in IL-2 or IP10 MDCs suggest that S4321 acts on the synapse between a T cells and an antigen presenting cells. So in summary, we think that S4321 offers a novel and differentiated approach over the first generation of PD-1 agonist antibody, which also are depleter antibodies. And we think that bifunctional activity has a potential for greater efficacies. And the bifunctional activity comes from a dual cell bidirectional antibody, a top hand, where PD-1 affinity mimics the natural ligand. By mimicking the natural ligand, we are avoiding drug-induced loss of PD-1 breaks, while the bottom end, being selective for Fc gamma receptor 2b, has no pro-inflammatory liabilities or does not deplete PD-1 positive Tregs by avoiding activating Fc gamma receptor. We signal the agonism not only through PD-1, but also to Fc gamma receptor 2b. So we simultaneously inhibit different pathways and different cell types. And with this, I would like to thank all the seismonauts that contributed to this program. Everyone here in these slides at one point of their life is seismic, really contributed to the realization of our DC candidate S4321. And I have the privilege of showcasing their work today. Thanks again for Eurofins for the opportunity of sharing our story, and I will be happy to take any questions.
Perfect. Thank you, Daniela, for that fantastic presentation. So now let's move on to the final part of today's webinar, which is the question and answer session. So as a quick reminder to our audience, you can still submit your questions using the questions panel in the menu on the right-hand side of your screen. So the first question we've got here is, one of the challenges associated with therapeutics with Fc binding Fc gamma R2b is an accelerated clearance by the liver. How is the PK profile of S4321?
First of all, this is a very good question. Here is our answer. So our Fc binds with low affinity Fc gamma receptor 2b. And the low affinity binding to Fc gamma receptor 2b is what prevents issues such as accelerated clearance, which has been observed with the use of higher affinity binders. S4321 actually demonstrated to be, the PK of S4321 demonstrated to be dose proportional. As we measure a 70% of bioavailability with subcutaneous dosing in cynomolgus monkey. So the PK is not affected by rapid or accelerated clearance, unlike other Fc gamma receptor 2b binder, but the difference is really affinity.
That's great. Thank you, Daniela. The next one here is, was the agonism abrogated by blocking Fc gamma R2b with antagonist antibodies in your in vitro culture systems?
Very good question. Yes, agonism was abrogated when we either use antibody that block Fc gamma receptor 2b, or we genetically delete, like I showed in one of the slides, Fc gamma receptor 2b by using CRISPR knockout. It was very interesting that the same was not observed if instead we block Fc gamma receptor 2a. So if we block Fc gamma receptor 2a, but we still have Fc gamma receptor 2b on board, we don't abrogate, we don't really change our agonistic potential. So this underlies really the role of Fc gamma receptor 2b in promoting PD-1 agonism or promoting superclustering in this case.
Perfect, thank you. This next question that's come in is, what are the mechanisms that explain the loss of PD-1 with high affinity agonism antibodies?
Correct. This is actually a very interesting question because we think that there are three mechanisms that can explain PD-1 loss. One is what I illustrated during the presentation, is PD-1 internalization, but it's not the only one. PD-1 loss of expression of PD-1 can be explained by depletion. So especially when we use an IgG1 wild type, this IgG1 wild type will mediate ADCC and loss of PD-1 positive cells. But the other mechanism is shedding, which has been reported. This is something that is definitely not the easiest to measure, but exists. And those three mechanisms can coexist. And the prevalence of one versus another, it depends really on the cellular makeup and the context that we are looking at.
That's great, thank you. This next question is, how did you use machine learning to support the selection of PD-1 clones deriving from canonical immunization approaches?
Yes, so machine learning actually allowed us to attempt the first in silico binning when we had our clones, during our selection of clones from antibody screening. Of course, we then verified also with orthogonal method, like with biosensor, the binning that came out from machine learning, and they agree. So machine learning was very useful, was able to predict the binds and the number of epitopes that we bind to PD-1.
Okay, that's great, thank you. This next one is, how did you determine the best combination of variable region and FC region?
Yes, this was actually probably one of the most difficult part of the program, because what we discover or reconfirm, it's not that we didn't know, that when we combine a variable region with a different FCs or vice versa, the result can be very different. So for instance, we discovered that the same variable region with different FC could be really efficacious or not, depending on the FC. So there is really an optimal combination of variable region and FC that we tested empirically by utilizing in vivo models of agonism. So this is really how we were able to tease apart differences between all our combination of variable region and FC region. So definitely was the most challenging part, in my opinion, during the discovery of the molecule.
That's great, thank you, Daniela. This one is, does PD-1 agonism disrupt binding and activity of natural ligand PD-L1?
Very good question too. So S4321 does not interfere with PD-L1 binding to PD-1. If any, we notice that when we use S4321 and then we add PD-L1, we actually enhance the binding of PD-L1. We believe that this enhancement is actually promoted by probably a change in conformation that we are inducing when we bind S4321. So in other words, the use of an agonist not only deliver itself an agonistic signal to PD-1, but also promote and enhance the negative signaling that comes from the natural ligand.
That's great, thank you. This one is, have you identified any potential biomarker of PD-1 agonism?
Yes, so we have observed a consistent upregulation of additional inhibitory receptors such as TIGIT on T cells when we are treating in vivo with S4321. And what this suggests is that PD-1 agonism can really lead to an increase of the number of breaks on T cells. So we do believe that that can be considered a biomarker of PD-1 agonism.
Great, thank you. So another question here, and this one is, was only one benchmark PD-1 agonist tested or multiple? And if it was multiple, did they behave the same or did they show any differences in your agonist reporter assay?
That is a very good question. I think there is a slide where you can see really the correlation between affinity and loss of PD-1 and how affinity correlates to the induction of L2, where we use different benchmarks. So we use 4 benchmarks that are characterized all by higher affinity compared to S4321, and they all behave the same in the sense that, of course, there is a correlation between affinity and their loss of PD-1, but the biology recapitulates.
That's great, thank you. So we've got time for one more question, and this one is, how are you approaching indication selection? Are there specific indications where Fc gamma R2b are more or less available?
It's a very good question too. So definitely you have to think that this drug might not work in condition where there is a B cell depletion, right? So B cells are important in order for us to agonize PD-1 because B cells express a lot of Fc gamma receptor 2b. However, in autoimmunity, this usually is not a cell line that is lacking. So in autoimmunity, we have an abundance of B cells. So we do not think that we will have any problem in seeing agonism because B cells are really present in autoimmunity. But both target needs to be mined in order to select the indication that you want to consider in the clinic. Is true.
Perfect, thank you. So we have got another question that's come through, and this one is, have you confirmed the PD-1 agonistic activity of your molecules in a humanized in vitro disease model?
That is a very good question. We did utilize some in vitro assays, and we did confirm some aspect of PD-1 agonism. But I think the best of the data, or the more granularity of PD-1 agonism in terms of cytokine production and the broadness, I would say, of PD-1 agonism was only observed when you run the molecule in vivo. So yes, in vitro assay can be very useful, but really you are going to see a very little picture compared to what you observed in vivo model.
That's great, thank you. So I think we've got a few more questions. This one is, which indication do you think is appropriate for S4321?
Well, we definitely, in general PD-1 agonism, our peers have proved clinical efficacy in array. So definitely that is a benchmark indication for also S4321. There are additional indications that we are considering definitely, but array is definitely the benchmark for PD-1 agonism so far.
That's great. Thank you so much, Daniela. Well, that's all we've got time for today. So thank you to our expert, Daniela, for a fantastic presentation and discussion. And thank you to everyone joining us online and for submitting your questions. And please note that this webinar will be available to watch on demand in just a few days' time. And as you leave the webinar, a short survey will appear on your screen asking you to rate your experience. So if you're unable to complete it now, don't worry, it will also be sent over to you via email shortly, and we'd greatly appreciate your feedback. On behalf of Drug Target Review and Eurofins DiscoverX, thank you for attending, and we hope to see you again soon. Goodbye, and thank you for joining us.
Thank you.
Thank you.